Hi, I'm Neil Mencken, and today I'm going to talk to you about a method for inferring biological networks by sparse identification of nonlinear dynamics. This was work done at the University of Washington in Seattle by myself, Steve Brunton, Josh Proctor, who's also at the Institute for Disease Modeling, and Nathan Kutz. The paper associated with this work is available on the archive at this link. So I'm very interested in using methods to identify the underlying model for regulatory and metabolic networks. So regulatory networks are, generally speaking, the networks that drive decision-making in cells. So if these decision-making networks break down, then this can lead to problems like paths to cancer, or developmental problems, or mitochondrial diseases. I'm also interested in metabolic networks, which are the networks that control all of the chemistry that's done by a cell. And in particular, I'm interested in how both understanding and being able to control these networks could lead to drug discovery or essentially hijacking the network in order to make different high-value products that we're interested in making in bacteria. So for example, biofuels. In order to both understand and control these networks, however, it would be very useful if we understood the underlying dynamics that are happening on them. And so model identification for these purposes means both understanding the network structure, so which components interact with which other components, and also what the functional form of the dynamics of those interactions are. So for example, the dynamics of x2 or the time derivative of x2 is as a function of all of the other components in the network that contribute to it. And we have hope that these type of networks could be inferred from data using a sparse methodology or a sparse regression methodology because generally speaking, these type of networks are known to actually be sparse. So not every component or every compound or every gene in the network is interacting with every other component. Generally, you can write down, if you can write down the dynamics of each component, it's a function of only some smaller subset of all of the different components in the network. In addition, these type of networks have a nice set of functions that can describe those dynamics. Generally speaking, these networks can be well described by mass action kinetics, and so the types of functions that we're going to be looking at on the right-hand side of these dynamic equations are polynomials in terms of those different functions. So for example, x2 or x1 squared or x1 times x2. And of course, we could be interested in much higher order terms depending on the cooperativity of the different components. Finally, once we discover, if we can discover the functional form of the dynamics of these systems, then we can also rely or then give these functions to domain experts, and they can rely on a broad amount of work that's already been done to understand the dynamics in terms of network motifs. So for more about network motifs, and this is basically the idea that a lot of the functionality of these systems happens in a modular way, in a reproducible modular way. So the same type of modules happen in many, many different systems. So for that, I refer you to Urielon's review in 2007. So in order to identify these underlying dynamics, we're going to work or build on a method that was published earlier this year by Brunton, Proctor, and Kutz in PNAS. And this sparse identification of nonlinear dynamics method, or SINDy method, begins with generating data from the system. So in this case, I'm showing simulated concentrations over time, so time series data taken at discrete points in time for x1 and x2, so x1 and x2. From this data, we're going to take this data and we're going to plug it into a library of nonlinear functions. So again, for the mass action kinetics in these type of biological networks, this functional space is going to be polynomials. And so we're basically saying that the dynamics of each state variable x1 and x2 will be equal to the sum of some functions in this space. So we take the functions, for example, 1, x1, x2, x1 squared, and plug the time series data into those functions and put them in the columns of a matrix. So even though the dynamics or these functions can be nonlinear, because we're plugging the time series data into those functions, we can still formulate a matrix that is just a matrix full of numbers. And so that's our function matrix or our data matrix theta. And in order to formulate a regression problem or formulate this set of equations, we say that theta times some other matrix C with the first column representing the coefficients for x1 and the second column representing the coefficients for x2, et cetera, is equal to the derivatives of the state variables. So also a matrix with the derivatives of the state variables plugged in. And you can get either measure the derivatives of the state variables directly or calculate them numerically from the time series data. So now we have a problem, we have a linear problem or a linear algebra problem that is a regression problem that's basically set up as our function matrix with our time series data plugged in. So that's our function matrix theta times a matrix of the sparse coefficients that are selecting which functions are active in the system is equal to the time derivatives. And we don't know, or what we would like to find is what the correct coefficients are. And we're going to rely on the fact that we know that the system is sparse. So most of those entries in Xi are going to be 0. And you can solve this sparse regression problem using either lasso, which is one of the most common techniques, or in the PNAS paper, my co-authors actually used an alternating method between least squares and thresholding. So they would solve the least squares problem and then threshold out some values in the resulting Xi vector that were below some value. Okay, so once they have found that those the sparse coefficients, then they can reconstruct and evaluate the identified model. So by identifying those coefficients, they determine which terms are active in the function library. So in this case, F2 is multiplied by an active coefficient, and F11 is also multiplied by an active coefficient for x1. So you can reconstruct the dynamics of x1 and then use test data for new initial conditions to compare the reconstructed model against new experimental, or in this case, simulated data from the original system. Okay, so this is the general framework for using a sparse identification of nonlinear dynamics method. Generate data, build a nonlinear library of candidate functions, and then formulate a regression to select which terms are active. And then finally, once you have those sparse coefficients from your sparse regression, then you can reconstruct the identified model and evaluate for new test data. Okay, so I was interested in applying this method to biological networks, and I'm going to show you 3 different networks, Michaelis-Menten, a competence network, and yeast glycolysis. And you can see one is just a single state variable, 2 state variables, 7 state variables, and these are all fairly well-known models within the systems biology community, which is why I selected them. And in addition, they present a new problem for the framework, which is that they all have not just linearly additive polynomial functions, but they actually have rational terms in the equations. And so Michaelis-Menten is not only one of the most famous models for describing enzyme kinetics, but it's also one of the simplest models that has a rational function and is relevant for biology. So again, this in order to describe Michaelis-Menten kinetics, you end up with the derivative of your state variable, or your substrate in this case, is equal, and so I have some influx of that times the reaction that's happening. And this rational function in this dynamic equation for the state variable arises very naturally because basically you have your substrate X binding to your enzyme, and this binding and unbinding is happening very quickly, whereas the actual reaction that produces a product is happening much more slowly. And so anytime you have this type of timescale separation where you can say part of the reaction is happening quickly and part of the reaction is happening slowly, you can reduce your system down to the single state variable by setting this the equation for these dynamics equal to pseudo-steady state, and you end up with a rational function. The problem, of course, with these rational functions is that there is not a simple way to formulate a compact library of them. So as soon as you get to two-state variables, trying to actually combinatorially write down all of the additive possible rational functions. So if you wanted to write, again, that different coefficients times each possible rational function, the library would get computationally impossible, essentially. So we need a compact way to formulate that library. So instead of writing x dot is equal to sum of functions in the numerator and sum of functions in the denominator, we're going to write it as an implicit equation. So multiply through by your denominator and move everything over to the left-hand side. And now we have, again, something that can be linear in terms of functions of polynomials, and also the derivative of the state variable is equal to 0. So we've taken the problem that was too hard to formulate a compact library for and turned it into a problem where we can't solve for the coefficients that would select for our different functions using the same method as before. And so now I'm going to show you how we got around that. Okay, so we have this implicit formulation, and again we're going to go and build a functional library from the data of the time series of the state variables and also their derivatives. And in this case now the derivatives are living in the library because we have some library that's representative of the functions times the sparse coefficients is equal to that right-hand side being 0. So again, we can use a library of polynomial functions in the numerator and a library of polynomial functions in the denominator times the derivative of a single state variable at a time. So whereas before we formulated this library for all of the state variables at the same time, now we're formulating it for one at a time, but it's still a function of all of the other state variables. It's only just a function of one of the derivatives of the state variables. Okay, so that's our new function matrix theta, and we now have this function matrix theta times our sparse coefficients equal to 0, and we're trying to find what is a sparse coefficient vector xc which satisfies this equation non-trivially. So we don't want that every xc is equal to 0. We want some non-trivial number or some non-trivial xc satisfying this equation. And we know that xc has to live in the null space of this matrix by definition. So we're going to calculate the null space. So that's saying essentially we know that xc is going to be some linear combination of the columns of the null space. All right, so now we've moved our problem into finding a linear combination of the columns in the null space that are equal to the sparse vector xc. So what we need is a method for finding a sparse vector in a subspace, and this problem, so finding a sparse vector in a subspace, has been studied, and we're going to use a method by Q, Sun, Wright, and Wright that's fairly recent, and it's an alternating directions method. So it actually does what we did in original SINDy, where it solves the least squares problem and then thresholds, and it does that. It does a soft thresholding and does that sequentially. So what it's actually doing is solving an L2 problem and then solving an L1 problem, regression problem. Okay, so a priori, we don't know how sparse xc will need to be in order to satisfy our equations. And so that's a free parameter within the method, is how sparse that is, and it's the threshold parameter we call lambda. So I'm now plotting the threshold parameter lambda on the x-axis and the resulting number of terms in xi from this alternating direction method on the y-axis. And you can see as you increase the threshold in lambda, the number of terms in xi drops. So as we increase that threshold, xi becomes more sparse. So from this, we can't tell. I've circled for you which xi ends up being the best xi for our system, or the correct xi, but from this, we still can't tell which threshold we should use for this system. So we're going to create a Pareto front, and the Pareto front is basically the error in how whether theta times xi is actually equal to 0 is equal on the y-axis with the number of terms as calculated here on the x-axis. And so at some, and you could use other error metrics. This is just the easiest error metric to use when implementing the method. But basically, as you increase the number of terms, the error has a sharp drop-off, and that sharp drop-off generally indicates the correct model. So again, I've circled which dot in that space ends up being the correct model. Okay, so now that we've found our sparse xi, we can take that sparse xi and multiply it by our theta matrix and reconstruct our nonlinear dynamics. So that xi had some terms in it that are from the numerator and some terms in it that are corresponding to the denominator. So we can take those and reconstruct the dynamics for whichever state variable we were looking at. Okay, and again, after we've assembled the inferred model, we would like ideally to perform a test with new data to see whether it's correct. And you'd have to essentially redo this, build the library of functions, calculate the null space, find alpha such that xi is sparse, and assemble the inferred model separately for each the dynamics of each state variable in your system. Okay, so now I'm going to show you an example, and this is for the Michaelis-Menten model. So this is a fairly simple model because it only has one rational function. It's only one state variable. So I generate two time series with different initial concentrations. So these are the concentration time series, and then these are the derivative, and I'm calculating the derivative numerically or directly from the system. So this is like assuming you had that data, but again, you could also calculate it from the time series of your state variable. Okay, so once we have this data, we build our nonlinear library. So since this system is only a function of a single state variable, the library is just the polynomial functions up to whatever order we want to go, and I think I only went up to order 3, so the functions are 1, x, x squared, x cubed, and so those are the functions in the numerator, and then in the denominator we need the same thing, so times the derivative. So x dot times x dot times x squared, and then x dot times x squared cubed would be the last one, if we wanted to use the same, up to the same order polynomial in the numerator and the denominator. So that's our theta matrix, and that's going to be multiplied by the sparse coefficient vector xi, and I'm showing you the values that the alternating direction method selected for the most parsimonious model, so that where that drop-off was on the Pareto front. And right, here's the Pareto front. So again, that's where the coefficients I'm showing you in that vector are the ones that occur, and it has selected 1, x. These two terms the coefficients are 0, and then x dot and x dot times x, with the rest of them also being 0. Okay, so we can use this to reconstruct the identified model, and you end up with x dot is equal to just a single rational function of polynomials. So in order to get back the appropriate model, we have to do some rearrangement. But again, if you are a domain scientist, maybe certainly getting this equation of a rational function is better than having to build the model totally from scratch. And if we do the appropriate rearrangement or factoring of that function, then we end up with exactly the dynamic equation that we put in to begin with. So it was a success. And of course, at the end, you would like to test your inferred model. Of course, since we know what the underlying equation is, we can look at this and say, not only is my structure exactly the same, but my coefficients are exactly the same. So of course, I'm going to produce time series that look exactly the same from both the inferred system and test data. But if you didn't, then that would be the appropriate test. Okay, so now I'm going to move on to the two-state variable regulatory network. So in order to perform the sparse regression for this problem and have it produce the correct set of equations, I had to use quite a bit more data. And in the future, we would like to understand how much data you need to use depending on the size of the system and the size of the library of functions in order to recover the underlying dynamics. I will say that when you don't have enough data or when you don't have the correct library of functions, then you don't get a good drop-off in your Pareto front. So it actually is fairly easy to tell when you need to either apply more, generate more data from the system or include more terms in your library of functions. All right, anyway, so this system has, again, two state variables, and so the library of functions in our function matrix that we're plugging our time series data into are now in terms of x1 and x2, so it ends up being a much larger data matrix, and of course, so it has the ones in the numerator and the ones in the denominator. Most of what I'm showing you here are the selected terms from the drop off on the Pareto front. So it selects x1, x2, those two are set to 0, x1 to the 5th, x squared, etc. And it's kind of hard to see, like I haven't shown you what the original model was, so I'll show you what that is. And so this is the original model. It actually has two sets of rational functions in each equation. And not only do we correctly find the structure of these dynamics, but we also correctly find the parameters or the coefficients A1, A2, etc. Within 2%. So you can again go and do perform a verification by taking new test data and comparing it against the inferred system. But assuming that you have the right amount of data, you can also recover this larger network. So the last example is the 7-state variable model, and again, I'm showing you the network. This one requires a lot more data. Unfortunately, at this point, I would say it requires a prohibitive amount of data. It's more than the amount of data that a biologist or a bioengineer would necessarily take on this network. But I still have confidence that this method could be usable. One of the things about this data is that I'm generating it using random initial conditions. So it's not actually the type of data that you would get from an actual biological experiment. And I think by using perturbation experiments or the type of experiments that biologists actually do, you could greatly reduce the amount of data that is necessary. Okay, so once again, we take this 7-state variable system, we build our function library of polynomials, we generate a Pareto front, find where the drop-off occurs, recover the coefficients, and we get back, again, here is the model, and here's the table of the true values of the coefficients and the extracted values of the coefficients, and once again, they are within 2% of the original coefficients. Okay, so I'm really excited about this method because it's data-driven. So you're going to get a model out that best describes the data that you give the method. It's also, and you're not going to get a model out if the information is not in the data. It also evaluates a relatively large class of functional forms. So if you think about other ways in which people are generally trying to determine the correct model for a biological network. Sort of the bottom-up creation of models can be very slow, and so this is, I feel like this is really a method for being able to construct or generate these type of nonlinear dynamic models very rapidly, given a pretty broad expanse of functional forms that can potentially go into them. The method's also relatively fast. Most for the 2-state variable and 1-state variable system, it runs on my computer in a couple of seconds, and for the large, and my computer is a laptop, and for the large 7-state variable system, it took it took a couple of hours, so it was a little bit longer, but it still wasn't prohibitively fast. So using this method, we correctly recover the network structure, the functional form of the dynamics, and the coefficients within 2%. For future directions, we're really interested in connecting how the sparse regression is selecting the functional forms that it is using an information criteria approach. And we're also interested in improving the method so there was no noise in any of the systems that I looked at, and especially the point where you're calculating the null space in this method can be incredibly sensitive to noise. So we're interested in improving that robustness to noise, specifically by using a method by Gavish and Donahoe. The archive paper is noted there. And finally, I'm interested in reducing the number of measurements using perturbation type experiments that are built into the regression or built into the functional library with control, so that we won't need a prohibitive number of measurements in order to recover a 7-state variable system. Okay. All right, well, thank you very much for listening. And again, more details of this paper, along with all the numerous citations, can be found on the archive. Thank you very much.